Hello everyone, welcome back to my channel Learn Biology in Easy Way. In this video, I am going to discuss about an important topic of microbiology, that is microbial growth. Microbial growth is defined as an increase in cellular constituents and may result in an increase in organisms' size, population, number or both. Microbiologists normally study the population growth by analyzing the growth curve of a microbial culture. The growth curve is usually obtained by cultivating microorganisms in liquid medium in closed system, that is batch culture. Batch cultures are those in which microorganisms are incubated in a closed culture vessels with a single batch of medium, and no fresh medium is added during incubation. Due to this, nutrient levels decrease and concentrations of waste increase. The growth curve of microorganism reproducing by binary fission is prepared by plotting the logarithm of cell number against the incubation time. On the growth curve, four phases have been identified. They are lag phase. When an inoculum is introduced into fresh culture medium, there is no immediate increase in cell number or mass. This period is called the lag phase. This phase necessary before the start of cell division because of various reasons. Cells may be old and depleted of ATP, essential cofactors and ribosomes. Composition of medium may be different from the one in which microorganism was growing previously. Microorganisms may have been injured and so require time to recover. The length of lag phase varies with the condition of the microorganisms and the nature of culture medium. The lag phase is long if the inoculum is obtained from an old culture or refrigerated culture. It is very short or absent if the vigorously growing exponential phase culture is inoculated into a fresh medium. Next, exponential phase or log phase. In this phase, microorganisms grow and divide at maximal rate depending upon their genetic potential. The nature of the medium and the environmental conditions, their rate of growth is constant. The microbes divide and double in number at regular intervals. The growth curve rises smoothly as each individual organisms divide at a slightly different moment. In this phase, the population is nearly uniform in terms of chemical and physiological properties. In a closed system, population growth remains exponential for only a few generations and then enters a stationary phase because of nutrient limitation and waste accumulation. In an open system, the exponential phase can be maintained for a long period by continual addition of nutrients and removal of waste. Next, stationary phase. In this phase, population growth ceases and the growth curve becomes horizontal. Bacteria achieve stationary phase at a population level of about 10 to the power 9 cells per ml, whereas other microorganisms like protozoa and algae do not reach such high population densities. They have a maximum concentration of about 10 to the power 6 cells per ml. The final population size depends on nutrient availability and other factors as well as the type of microorganism. In this phase, the total number of viable microorganisms remain constant either due to a balance between cell division and cell death, or the population ceases to divide. Population growth in this phase ceases either due to depletion of nutrients or due to accumulation of waste. Next, death phase. In this phase, depletion of nutrients and accumulation of waste in the culture medium lead to decrease in number of viable cells. In this graphical representation, we can see logarithm number of viable cells in y-axis and in x-axis time. In this curve, this part of this curve show lag phase, this part of the curve show exponential phase, and this part of the curve show stationary phase, and this part show the death phase. Thank you.